My name is Nolte. I'm a microbiologist by training, although I'm a scientist. I've studied biology. At this time, I'm the head of the diagnostic department at the University of Zurich at the Institute of Medical Microbiology. So I'm responsible for the bacteriology unit, for the molecular, for NGS, and for microbiology. So to briefly characterize the IMM, it's a laboratory which serves the University Hospital of Zurich. We have approximately 70 employees in the diagnostic unit, a number of academic specialists, and a number of technologists. So if we talk about the impact of AI in the clinical microbiology laboratory, which could impact research projects as well as the diagnostic routine, then we have several points which are of interest or which can be impacted by AI. If we start with the routine work, where we use AI applications for pre-classification of cultural growth, for instance, from urine, then we have impact on first the time to result, which is shortened for those samples which don't show growth or which have, let's say, clinical insignificant growth, because they can be processed with, let's say, just one mouse click in a batch. We have an impact on standardization, which is, to my point of view, very, very important, because the interpretation of the growth from a clinical sample, from a urine, depends to a large extent on the knowledge and expertise of the technologist in front of the screen. So it could be that one sample is categorized differently by 2 or 3 technologists, and that's not what we want. We want to have some sort of standardization, which allows that the interpretation of growth depends on the same criteria. And this is where AI can help us pretty good. If we take a look at the research side, what can we expect, or where can we use AI maybe in the future? There are different topics we are working on. So one thing is to use, let's say, large language models somewhere in the routine diagnostic process, be it for the management of SOPs, for quality assurance, quality control, be it for answering questions within the laboratory or outside the laboratory. We are working on projects to pre-classify images from culture or from microscopy in order to get more deep interpretation as compared to what we now have with the phenomatrix. As a service laboratory, we report our results. We are in contact with the clinicians. We have a dialogue with the clinician, but on an individual level, it's very hard to rate whether we have a positive impact. It has been published several times that with the use of artificial intelligence and automated processing of samples which have no growth or insignificant growth, you can have a reduced time to result, which then has an earlier actionability for the clinician. So the clinician, the waiting time for the clinician to get an actionable result is reduced, even in those cases where we don't find a clinically relevant pathogen. So if we think about a broader impact of AI applications, machine learning, or software which is based on both for the patient itself or for the diagnostic process, I could imagine that with the interoperability of data, which we don't have yet in a sufficient way, we might be able to develop patient-centered diagnostics. This would mean, from my point of view, that we not only have a look on the laboratory results in the bacteriology unit, so identification AST, but that we have results available from other diagnostic units as well as from the clinics, so that we can integrate the different classes of results to have a more precise interpretation of what we are seeing in our cultural approach. If we interconnect our microbiology laboratory with the clinical information system, with the ward like an ICU, we can probably integrate data from both the lab and the ward to get this complete overview for a patient, which would enable us to better interpret the cultural results, which would enable us to do a susceptibility testing in a more personalized way, because we know the pre-treatment of the patient, we probably know other conditions which are relevant for susceptibility testing, and by this we could contribute with a real useful report much earlier than we do it today. So if we talk about artificial intelligence or software which uses AI in the laboratory, we also need to talk about training of medical experts, of technicians, of the staff in the laboratory. And here I see a big advantage by using the digital bacteriology or microbiology which we are approaching, because we can use specific cases where we have the complete digital record, not only the patient record, but also images of the plates. We have the knowledge on how those plates have been rated, for instance, by Phenomatrix or other artificial intelligence applications. And so we can use those really good defined cases in teaching students, in teaching trainees which come to us during the education to become a medical technologist. And we can also use those digital cases to align with other laboratories, to compare if for the same sample type or for comparable conditions, different labs may come to the same conclusion, which is not always the case at this time. It is pretty interesting to try to draw a picture how the microbiology lab might work in, let's say, even 5 years or maybe 10 or 15 years, and try to imagine what kind of technological changes might come to support us or to change the microbiology as we know. Bacteriology in itself is a very, let's say, traditional discipline in diagnostics, and it's currently undergoing a revolution. Revolution started with MALDI, then we got the streaking automated, then we got the automated laboratories. Now we have first applications which are based on AI and machine learning, and all this in very few years. And I guess that this is just a starting point. So we are playing around a little bit with the possibilities which machine learning gives us. If I would try to imagine what is the situation in the next 5 or 10 years, I could envisage that that we improve those existing applications, but that at the same time we much more integrate data from other sources to get the holistic view. This is probably something we will approach within the next 10 or 15 years. Another thing, what excites me is the vision of having an automated process which starts with the inoculum to the final outcome of culture. So the need for identification, the need for AST is completely done in an automated way. I think this is a way of working which we could approach within the next 2, 3, or 5 years. It depends a little bit on the technology which we are using. But this completely intervention-free process. And then there's a third thing which is pretty exciting to me and which I'm following for many, many years now, at least in theory. This is the question: would it be possible with standardized culture to get at least a pre-identification of the most relevant pathogens without multitoph? So at this time in the laboratory, we get the culture, we do a picking, we subject the biomass to multitoph to get the identification. But the trained human, the trained microbiologist, for instance, is able to pick up a blood plate and to say, okay, well, this is Staphylococcus aureus, this is a Streptococcus. So the same should be possible with the use of machine learning by analyzing the standardized cultures which we now get. And this has been published last year, that some groups are able to identify up to 30 different pathogens from urines without multi, just by the appearance of the colonies on the agar plate. And this is fascinating. So I could imagine that at a certain time in 5 or 10 years, we not only have the image of the culture, which we have right now, but that at the same time, when the image is been done in the smart incubators, we get a report on which bacteria have been detected on that culture plate in which quantity. And if we then integrate with data which we have from the clinics, then we could even have an interpretation of the list of pathogens which have been identified in terms of clinically relevant or not. So this is the last aspect, is what really excites me, and where I think with the use of standardized microbiology and powerful machine learning projects, we may approach a completely novel workflow in bacteriology, which is really important if we talk about software and algorithms in general. So AI, machine learning, all these applications and possibilities and chances that we have. One aspect is the regulatory aspect, which I personally see critical. We need regulation for medicinal products. Of course, there's no doubt about this. We need it to ensure that we have really control on what is being released to the diagnostic market. But at the other side, the regulatory authorities probably not fast enough to follow the technological progress which we see. So at this time, if we use or if we want to use applications from our research department in routine, this will be a laboratory developed test which need to be validated by us in a very critical way. And the question, the machine learning program can be validated according to the current IVDR regulations is not answered, at least to my understanding. So with the progress of artificial intelligence in the clinical microbiology laboratory, we need a critical discussion with the authorities to enable that also AI software or AI applications can get the label that they are IVDR conform in a meaningful way. And this is a sidekick of what we are seeing in the microbiology laboratory. We see a lot of progress, at least in theory, at least in research projects, but we are hampered by regulations which don't really fit to artificial